there's no way that I'm going to be able to do total justice to this broad category. So what I'm going to narrow it down to are talking about diseases of amphibians, reptiles, birds, and mammals. And you'll notice that I'm going to put a lot of emphasis on amphibians and reptiles because I think this is probably about the only place you're going to get that kind of information. A lot of the diseases we see in birds and mammals are analogous to what we see in domestic animals. So you can extrapolate from what you know about domestic animals. But the amphibians and reptiles, that's not the case. Oh, thank you. So without further ado, I will start off talking about diseases of amphibians. Now, the general scheme of my talk is I'm going to talk about infectious diseases, and then being that I'll talk about viral, then bacterial, then fungal, then parasitic, and then I'll move into other kinds of diseases. So if you're writing this down for some reason, that'll kind of be the scheme. The class Amphibia is divided into 3 orders, one of which I'm not going to talk about at all because they consist of blind, legless creatures that live in the soil in the tropics, and no one really keeps them in captivity. The other 2 groups are the class Anura, which consists of the frogs and the toads, and the class Caudata, which are the tailed amphibians, which are the broad group of salamanders. All right, the first disease I'm going to talk about is a disease that's caused by a herpes virus. It's known as leopard frog herpes virus, or the Luque's herpes virus. It was so named for Dr. Luque, who described it back in the 30s. That's L-U-C-K-E with the accent over the E. What we see here in this leopard frog is there's some abdominal distension, and this is because there's a lot of ascites because this animal is in renal failure. And the slide conveniently shows us what is left of the right kidney. The rest of the right kidney has been replaced by a large nodular mass, and the entire left kidney has been replaced by this mass. This is a neoplasm that is virally induced. It was one of the first neoplasms that was proven to be caused by a virus. And the interesting thing about this is that when this was first described back in the 30s, there was a lot of excitement about it, and people went out and they caught up a bunch of the northern leopard frogs, and they found that there was an incidence of about 10% of the adult frogs had this tumor. But they depleted the population so much that after a few years, the incidence dropped way down. And this is definitely a disease that's related to population density. The more dense the population, the more chance there is for the virus to be transmitted between the animals. Histologically, I'm not going to show a lot of histo at this, but I just wanted to point out it is a adenocarcinoma of the kidney, and at certain times of the year, you can see the nice intranuclear inclusions. These are only seen during the winter and early spring months. This is a temperature-dependent phenomenon. And it's kind of an interesting strategy if you can think of a virus having a strategy. The virus active viral production occurs in the winter time when they're hibernating, and in the early spring when they first come out of hibernation, so that when they get together for their spring matings, there's lots of virus within the kidneys and within the urinary bladder, and it's being excreted into the environment. And the animals become infected as eggs or as early tadpoles. And during the summer months of the first year, there's no viral production, but there is tumor production. So that by the end of the fall, just before the animals go into hibernation, there is a tumor present, and then they hibernate, then there's a chance for the virus to replicate, and the following spring, then the virus is passed on to the next generation, and then the frogs usually die during the second summer. So, kind of interesting. Whoops. Well, that's pretty much it for viral diseases of amphibians that you're going to have to know about. The next thing I want to talk about is bacterial disease, and what we see here, notice this is a red-eyed tree frog, and there are multiple areas of dermal necrosis and hemorrhage. This is due to bacterial septicemia, and it's a very non-specific change. It can be caused by any kind of bacteria. It's usually caused by Aeromonas hydrophila. I say usually because that's part of the normal flora of the skin of these things, and it's in the environment. And under poor management conditions, overcrowding, poor water quality, stress for other reasons, then the bacteria can replicate to high numbers and overwhelm the animal's immune system, and then you get things like this. As I said, it's often stress-related, and this is the same frog opened up, and all these blue things are eggs. And we see this a lot around the time of egg laying, and I think that just you can appreciate the sheer amount of energy and effort this animal's putting into reproduction, and that's got to be stressful. So often we see it in animals that are getting ready to go to lay eggs or to mate. The more common name for this disease is red leg. You probably know about this, and this is one of the diseases that anybody who's taking any kind of upcoming boards probably should know, because they don't ask many questions about amphibians and reptiles, but this is a classic. Show a picture like this, you'll see the petechial to ecchymotic hemorrhages of the skin, and the answer, if they want to know the syndrome, it's red leg or bacterial septicemia, and the most common agent causing it is Aeromonas, although just about any kind of gram-negative organism can do it, E. coli, Citrobacter, etc. And it's also been, we've seen it due to Staph aureus, and there was a report out of South America in bullfrogs where it was caused by strep. So really, it's under the right conditions, just about any bacteria can do it. Usually this may be all that you see. It's just this acute septicemic process and the animal dies. If it lives a little bit longer and there's time for lesions to develop, or at least gross lesions to develop in other organs, often you'll see areas of necrosis in the liver, as we see here. You can see there's white foci, multifocal necrotizing hepatitis, and even in the heart here you can see there's some muscle necrosis. You often see areas of necrosis and septic thrombi microscopically, but as I said, the animal has to live a little while before you're going to see it grossly. This is an axolotl, which is a Mexican salamander, showing a large area of dermal ulceration. Not all bacterial infections have to end up as red leg or acute septicemia. Often, if an animal is traumatized, you can get secondary bacterial infections, and you end up with a large area of ulceration, such as we see here. If you culture this lesion, I guarantee you'll get Aeromonas out of it and probably a bunch of other things. And the question is, did it cause it, or is it a secondary secondary invader? And that's usually the case. It's secondary to some other problem. In this case, we were pretty sure it was trauma-induced. I throw this slide in. This is a common laboratory animal that I guess everyone knows what it is. It's the African clawed frog, Xenopus lavis. This is a normal Xenopus lavis. Well, normal except it's dead, but it did not die from any particular disease. But I throw it in here to remind me to mention that there have been several reports of acute septicemia in Xenopus, laboratory Xenopus, due to chlamydia psittaci. And I'm aware of at least 4 reports now. So if you're working in a laboratory situation and you've got a sudden die-off of Xenopus, and the gross lesions are the same as red leg. I mean, petechial ecchymotic hemorrhages, liver necrosis. But you better keep that in the back of your head, because if you're opening these up, you might want to make sure you've got masks and gloves, and maybe even do it in a hood, because it is a zoonotic disease. Not all bacterial infections are acute. Some of them are chronic, such as we see here. I think you can appreciate, looking at this liver, there are multiple nodular areas. You can kind of tell by the shape of these things that they've been growing and coalescing over a period of time. Perhaps when you cut into this, you definitely could appreciate at that point that there's probably fibrosis surrounding this. This is due to mycobacterial infection. Amphibians do get mycobacterial infections. We've seen it lately. We've seen it quite a few times at the National Zoo. The most common organism that you're going to isolate is going to be some kind of waterborne saprophyte, such as Mycobacterium chelonii, morinum. We had several cases due to Mycobacterium gordoni. You're not going to see M. tb or M. bovis out of these things. Moving on to Fungal diseases, there's a condition that occurs in fish and in aquatic amphibians known as Saprolignia or Saproligniasis. It's the causative organism is broadly named Saprolignia, although I guess that was originally based on finding one species in the genus Saprolignia that caused it, but really Saproligniasis can be due to over 20 different species from numerous orders and families, and the general term just is to call it Saprolignia, although technically that's not correct. Again, this is a fungus that's associated with poor husbandry conditions, usually cold water temperatures. The animals are probably stressed, not eating, kept in below optimum water temperature, and the fungus then opportunistically infects the skin or the gills, as we see here. These are in the, who's doing that? These are in the gills, and it often will infect an area of trauma in the skin. The classic gross sign or gross appearance is a cottony white growth when you see it underwater like this. Now, I've got to be honest with you. I didn't have a good picture of Saprolignia, so this is actually cotton that I've wrapped around the gills of this thing. I do have one picture that truly was Saprolignia, but this is very typical. When you take the animals out of the water, the fungus kind of collapses down upon itself and often will just fall right off. This is the lesion that was attached to this gill. You can see it's a very superficial infection, but it does cause an ulcerative well, what an inflammation of the gill would be ulcerative bronchitis. Or you could probably get points if you just called it ulcerative dermatitis in this case. But if you took this mass of goo and put it under a microscope, do a wet prep, this is the kind of thing you're going to see. It's just lots of fungal hyphae and spores and that kind of thing. The best way to treat it is just warm the water up. That's bring it up above 20 degrees Celsius, and that'll kill off the fungus, and the amphibians will survive. There's another condition that we've seen a lot at the National Zoo. This is ornate horned frog, and you can see these things normally have a lot of cutaneous nodules, but if you look up near the nose here, you can see that there's ulceration, and these nodules are brown as opposed to these green ones back here, and you look down on the legs, it's the same thing. This is due to an infection with another, again, it's a broad classification of fungi called chromomycosis or chromoblastomycosis. Both terms have been used. The name chromoblastomycosis comes from the fact that they are naturally pigmented fungi. When you don't have to do any special stains to see them. You can just do a smear and you'll see these brown pigmented fungi under the microscope. They are, in our case, we've seen it in 2 or 3 different species of frogs, and it went systemic. And as you can see here in the liver, multiple areas, multiple granulomas, granulomatous hepatitis. We literally have seen lesions in every organ possible. We've seen it in the brain, in the bone, in the gonads, just everywhere in these. And these are, again, saprophytic organisms. They live in soil. I guess in a laboratory situation, you don't have your amphibians on soil, so you're probably not going to have a problem with this. But of course, we're trying to keep them in a natural environment for display purposes, and it's been a big problem in a couple of our exhibits. Now, in the literature, they talk about it being more of an ulcerative lesion. This is a leopard frog from Michigan, University of Michigan. You can see these large areas of ulceration, and you might say, well, why isn't this red leg? Well, I mean, look at the margins of these lesions. You can appreciate that it's a more chronic process. They're raised. It's not just necrosis and sloughing. There's been some time for this to ulcerate and expand, and that's what the classic original descriptions of it were, that it was ulcerative. Where it's been more of a nodular proliferative type of process. And again, the literature says, well, the causative organisms are fairly easy to culture, and that has not been our case. We've never been able to culture it. So we're probably talking about a totally different kind of fungus that, because it's pigmented, we group it under the name chromomycosis. All right, moving on to parasitic diseases. This is a Suriname toad, and you'll notice that there are multiple nodules on the serosa of the gut, which has been reflected craniad for some reason. And these are the ovaries down here, but these large nodules are developing ova, but all these little nodules are not normal. And if you think back on your veterinary school days and parasitology, you'll remember that they talk about a lot of parasites, that the intermediate hosts are amphibians and reptiles. Well, that's certainly true. And it is, I would say it would be unusual to rare for a wild-caught amphibian not to have some encysted parasites within the body cavity or within the muscles. This particular parasite is a Cess Toad. These are our larval Cess Toads that we were able to get them identified, and it turns out that these were a Cess Toad called Brevi multisecum, and the definitive host is a caiman. These are South American frogs, toads, and they come from areas where there's a lot of caimans, so I take it that caimans must eat these, because every single Suriname toad we've ever opened up have had a lot of these encysted parasites. The next picture just shows you a little closer view showing these things. They're very benign. They don't cause the amphibian any trouble, but when you open them up, you may be taken aback by the presence of these things. And they're not only host to cess toads and trematodes, but we also see a lot of encysted nematodes as well. And this is a smoky jungle frog, and these are I don't really know what nematodes these are, but they're probably ascarids of a bird or a reptile or maybe even a mammal. Again, a very benign process. They're just sitting there waiting for this amphibian to get eaten by a predator. Now here we have this is a African clawed frog, and you can tell that there's an excess mucus on the skin, and I hope you can appreciate the skin is somewhat reddened and has kind of a granular appearance, and that's because it is irritated due to infection with a parasite. It's a capillarid parasite that lives in the skin of these things. I think the name that is most commonly used now is pseudocapillaroides. It's pseudo and then capillaroides, like capillaria. Xenopi, although I do know it was formerly called Capillaria xenopodus. But as I said, I think the name that's recognized now is pseudocapillaroides. The life cycle of this is not well understood. I'll show you a histo. No, I won't. I took that out. I guess I took that out last night when I was trying to cut things down. But anyway, again, it's a very superficial infection. The parasites occur within the epidermis. They do not penetrate down into the dermis. And when you look at the skin histologically, you can see tunnels where the parasite has been tunneling through, and then with associated inflammation. And if you're lucky, you'll get cross-sections of the parasite or typical capillaria-like eggs, the bipolar plugs. I thought this was kind of a incidental thing that, yeah, well, it's been reported, but it's not seen nowadays. But apparently, it's still a very common problem in laboratory situations. And I don't understand, because you think they'd be able to clean them up better in labs, but I guess the life cycle must be direct. And that no one's really proven this, but given that in a lab, there's probably not much chance for intermediate host to occur. And it can be fatal, because the mucus production is an indication that the animal is irritated and stressed, and they can go off feed, and then secondary bacterial infections or fungal infections can come in on where these parasites have burrowed and lead to death. In addition to being the intermediate host for nematodes, they do have their own internal nematodes that they carry. This is the lung of a frog that's been opened up, and these little black squiggly things are lungworms. The genus is Rhabdius, R-H-A-B-D-I-A-S. There's several different species that can occur. These are, again, very common in the kind of frogs that I deal with, and that is leopard frogs, ornate horned frogs. I don't believe that it's been reported in Xenopus, but these are rhabditiform type parasites. They do have a direct life cycle, and it's very common to see these. They're usually considered to be innocuous infections, although I have seen infections heavy enough that it killed the animal. And the larvae are coughed up, swallowed, pass out in the feces, and then infect the frog by penetrating through the skin and migrating through body cavities before eventually ending up in the lungs. So you can see if you've got a cage situation where you're not changing the substrate very frequently, that there is a chance for these things to build up to high numbers, and then that's when you start running into problems, either due to the adults in the worms or the migrating larvae. All right, well, that pretty much takes care of the infectious diseases. Moving on to nutritional problems. There haven't been too many nutritional problems reported in amphibians, but it is recognized that they can get metabolic bone disease, and associated with improper calcium, phosphorus, vitamin D ratios in the diet. And this is one of a group of smoky jungle frogs that we were very proud to have gotten to breed and hatch and metamorphose into little froglets. But then apparently there was something in the diet that we were giving them, which I think was probably mostly crickets when they first metamorphosed, that didn't have enough calcium, or at least had an improper balance of calcium and phosphorus. And what we found was a lot of these guys had folding fractures of the long bones, as you see here in the femur and down the distal tibia. They seem to manage quite well. This is not what killed this animal. These were just found at necropsy, but these were kind of healed fractures here. But metabolic bone disease has been reported in other colonies of frogs as well. And again, it's usually because they're in a situation where they're raising them from the egg stage on, and there must be something missing in the diet. And another condition that's been seen in a number of frogs, mostly tree frogs, like this is a Cuban tree frog, is this corneal opacity due to deposits of cholesterol within the cornea. This is lipid keratopathy. And of the reports that have been in the literature, I think in every case it's been in tree frogs that were given diets that consisted mostly of mouse pups or pinkies. And the theory was, well, in the wild these things are eating insects which are kind of low in the cholesterol, and you're giving them these mouse pups with milk in the stomach, and you're probably just overwhelming their system. Now this has never been proven, but it sounds like a good theory to me. And sometimes this deposition of cholesterol can be quite extensive, leading to virtual blindness of the frog. I mean, this animal, there's no way it's going to be able to find food to eat it. The only way to keep it alive is to force feed it. So that's lipid keratopathy. Not proven to be nutritional in basis, but that's most likely pathogenesis. This is an African bullfrog, and what you see here is this is not his tongue sticking out. This is actually a mouse, and these guys are very attuned. Amphibians in general, most species, you got to give them live prey. Now, Xenopus is an exception. You can get the Xenopus to eat frog brittle. But most amphibians, you've got to have moving prey. And that moving stimulus, I mean, it's kind of hardwired. If they see something moving, they grab it. And they don't know when to stop. In this case, this animal, I mean, you can see he ate one mouse pup, and then he kind of went for an adult mouse, and his stomach ruptured. And he couldn't even get all the mouse down. I guess the tongue was sticking out for a few days, and then the animal died, and it had a ruptured stomach. Just due to pressure necrosis from this large amount of food in the stomach, and also the presence of the stomach filled with food makes it difficult for him to breathe, because the trachea opens up right here and bifurcates into the bronchi, so that it's kind of partially blocking the bronchi. This is a lung filled with air up here. Another problem, you got to watch out to make sure you keep your animals of different sizes housed separately. Again, as I said, they are very much stimulated by movement, and in this case, These were a group of different aged frogs that were kept in the same cage, and they dumped in a bunch of crickets to feed them and came back a little while later and found this situation. We assume that they probably both went for the same cricket at the same time. And the big debate here was, do you give this one 324 or do you give this one 324? Who died first? This is another Suriname toad, and these guys have an interesting feeding behavior. They and Xenopus do not have tongues. Some people argue then that you should even put them into a category outside of frogs and toads, and other people, the splitters say do that, the lumpers say no, they're just tongueless frogs. But anyway, their method of capture is they wait for something to come by, and then they just open up their mouth and kind of suck it in like a vacuum cleaner effect. And this was a case where we had these housed in an aquarium where there was gravel substrate on the bottom, and put some worms in for him to eat. And I guess he went down to grab a worm, and he sucked up a rock as well. And you can see this rock penetrated through the stomach. You see the hemorrhage here. So we've also seen problems keeping them on sand, the same thing. They just kind of suck up that sand, and then they get impacted. So I guess if you've got Xenopus, you shouldn't have rocks in the aquarium, or at least you have big rocks, so they can't suck them up. Okay, well that pretty much does it for amphibians. I'll move on now to reptiles. The order Reptilia, class Reptilia is divided into 4 living orders. The first one are the Tuateras, the Rhinocephala, which there's only one zoo in the country that has them, and they're only native to the north of New Zealand, and really nobody has them, and I don't know anything about them, so we're not going to talk about them. Then the other 3 orders are the Crocodilia, which has the crocodiles, the caimans, and the alligators. Then the order Chelonia, which has tortoises, turtles, and terrapins. And the order Squamata, which is subdivided into the sauria, or the lizards. This is a rhinoceros iguana here. And the serpents, or serpentes, the snakes. And before I go further, I thought I'd maybe, for those of you who aren't used to looking at the internal anatomy of snakes, they are somewhat unique and modified for their unique elongate structure. So I thought I'd give you a brief anatomy lesson here. This is a snake that's been opened up. His head would be up at this end. This is the heart. Here's the lung. Most snakes only have one lung. It's kind of an adaptation. If you think about it, if they had to squeeze 2 lungs into this space, the lungs kind of have to compete with each other for space to breathe. So they've adapted by making one lung that's very long. And some snakes, the lung tends to attenuate as it goes down towards the cloaca and then end up in an air sac, a thin-walled air sac. And in this method then, when a snake breathes in, it's able to bring oxygen in and fill up the airways up here in the lung parenchyma, but also fill up the air sac with air. And then as he breathes out, fresh air passes from the air sac back through the lung, so you can get it's more efficient oxygen exchange, kind of like what birds do. Then, as I said, everything is stretched out and elongate. So here's the heart, here's the lung, here's esophagus coming down here. This is probably the beginning of the stomach here. And this is liver up here. The liver is very long. And one interesting thing is the gallbladder is not up with the liver. The gallbladder is down here by the pylorus. This is stomach. I guess this is the pylorus here, your junction to the duodenum. And this is a very good anatomic site. You find the gallbladder, and then you'll find the spleen and the pancreas. In most snakes, the spleen and the pancreas are fused into one organ called the splenopancreas. In the boas and pythons, they are actually separated. There's a separate spleen and pancreas, but they're all still right here together, kind of in this little triad. And then the intestine in general is not very long, which is not surprising since these are carnivores, strictly carnivores, and the intestinal tract of mammalian carnivores tends to be short. And then you end up with, this is gonad here. This happens to be a male testis. Then the kidneys are lobulated and on each side, and again, elongate. So that's a brief anatomy lesson. The lung, a normal lung, this lung has been cut and opened up so that it has a central mesobronchus, and then off of that there are outpouchings, which technically should not be called alveoli, but a lot of people just call them alveoli. Anyway, they're air spaces, and that's where oxygen exchange occurs. So that's what a normal lung looks like. Now, the first disease I want to talk about is a viral disease. It's Paramyxovirus. And this is a virus that was first reported back in the 70s as causing an outbreak of pneumonia in a captive collection of vipers over in Switzerland. And this is, since has been reported in Europe many times and in this country many times as well. It's primarily a disease of vipers, poisonous snakes, but it can get into other groups. And it most commonly occurs in outbreaks where they've got vipers that they're keeping for venom production, or in a zoological situation where we've got a bunch of different species of snakes. It's assumed that there's probably some carrier snake out there, but nobody really knows what it is. There's different theories about it. But anyway, what the virus does is, if it's like Paramyxovirus and most other species, you get a nice interstitial pneumonia with marked thickening of the interstitium due to inflammation, and then there's proliferation of the epithelial cells, respiratory epithelial cells. It looks a lot like measles or distemper in mammals. And then these air spaces fill up with exudate, and the animals literally just suffocate. So this is a interstitial pneumonia due to Paramyxovirus. This is just to show you what a normal histology looks like. This is a normal snake lung. You can see this is the central mesobronchus, and then these outpouchings where the oxygen exchange occurs. Yeah, okay. I thought I'd taken that slide out last night. But anyway, what happens is this interstitium becomes infiltrated, and with Paramyxovirus, the interstitium becomes infiltrated with inflammatory cells, and then there's marked proliferation of the respiratory epithelium, so that these septa become thickened, maybe up to 10 times as thick as they normally would be. And then the animals just can't get oxygen across the membranes like they normally would. Plus, then these airways fill up with exudate. Okay, the second viral disease I want to talk about is crazy boa disease. Actually, that's not the name. It's called boid inclusion body disease. And it occurs in pythons and boas of many different species. What this boa is showing is neurologic signs. It's not normal for a snake to, I don't know if that's stargazing. He's kind of gone beyond stargazing. He's looking back down at the ground again. But in most cases, it's an asymptomatic infection. It's kind of something you discover at necropsy. You see the typical inclusions. It's called boid inclusion body disease because here we have kidney, and you can see in the renal epithelium there's these giant eosinophilic inclusions, intracytoplasmic inclusions, and that's where the name came from. You see it in the renal epithelium, you see it in the pancreas, bile ducts. Those are the most common places to see it as a purely incidental finding. When it does cause clinical disease, it's usually a neurologic disease, and that's what we're seeing there with that boa. It can go to the central nervous system and lead to derangements of neural function, often without any inflammation. Now, it seems like in pythons, inflammation is more common, but you can see it in boas, just these inclusions, and apparently it's enough to alter neurologic function so that they are showing neurologic signs. Now, what kind of virus is it? That's the $64,000 question. I guess the most recent thinking is that it is a retrovirus, although there's a lot of debate about that. Originally, they thought it was a reovirus. Now they think it's a retro. I haven't seen any definitive publication on it, although what I have heard and seen about it, I guess I do believe it's probably a retrovirus. And they think that it's carried by mites and other blood-sucking arthropods. Again, I'm not sure it's been proven, but it seems like you can take an animal that's clinically affected, put it in the same cage as a normal animal, and the normal animal never changes, but if there's mites involved, then you can transmit the disease, at least anecdotally. So anyway, it's kind of an up-and-coming disease, and I'm sure that there's going to be some articles published soon, hopefully where they say what it is. Okay, moving on now to bacterial diseases. This is a Australian beaded lizard, and it gets the name beaded for these things, not for these things. These are abnormal areas. You see there's a proliferative dermatitis or epidermitis, hyperkeratosis, and this is due to superficial infection with Dermatophilus congolensis. This was originally described by my boss, Dr. Montali, back in 1975, and since then it's been described in several other outbreaks. Often, again, it's associated with poor husbandry. The animals stressed. These animals came in from Australia. I think they were on a slow train. It took them a couple weeks to get from Australia to the zoo. They weren't kept at optimal temperatures. The cages were kind of moist and not real clean. And anyway, a lot of them had these superficial infections, and they are quite treatable. Again, you warm them up, you improve their nutrition, give them some antibiotics, you can clear these up. But if you don't, I mean, they can progress on and actually kill them due to secondary infections. So anyway, that's dermatophilosis. This is a black rat snake, and what I'm trying to show is these blisters. See these raised areas? This is called blister disease. It's the common name for it. They're actually vesicles in the superficial skin. And again, this is usually associated with poor husbandry, often keeping the animal under too moist and too cool conditions. And you can get superficial bacterial infections, and then what happens is these blisters rupture. It's upside down, but that's the ruptured blister, same blister that I squeezed. And you can see the clear fluid in it. What happens is these things pop, and then bacteria and fungus gets in there, and it can lead to ulcerative dermatitis, like we see here. And this is the ventral scales of a mangrove snake, and this is one of the most common areas to see it, because these other scales aren't real happy either. You see how they're curled up like this? This is an animal that's been kept in a cage where its belly has been moist all the time, and there's a lot of, moist and dirty, and then they get ulcers. And what kind of bacteria are you going to get out of here? Well, I guess the most common bacteria on the skin of a reptile is going to be Pseudomonas aeruginosa, although again you can get Aeromonas, a lot of gram-negatives, E. coli, Citrobacter, whatever. There's arguments that perhaps this is more of a phenomenon of captivity. There's been cultures done of wild snakes, and they say the flora in wild snakes is totally different, that maybe it's because we keep them in captivity and keep them under kind of unclean conditions, and it lets the Pseudomonas and the Aeromonas colonize the skin and then kind of wait for something like this to happen and then cause disease. Anyway, debatable. Okay, this is a mata turtle, and you can see what the mata is. The large area of ulcerative dermatitis. This is analogous to remember the frog slide I had with acute septicemia and ulcerative dermatitis? Well, the same thing in this turtle. When this was originally described, there was a report of it, they called it Septicemic Cutaneous Ulcerative Disease of Reptiles, or SCUD for short. Not to be confused with Scud missiles. But that name's kind of fallen out of favor. I mean, that was invented back in the 60s as a cute little acronym. Really, it's just, if you see this, it's not due to any particular organism. It's usually gram-negative infection, but you can have gram-positive infections, and it's just a sign of septicemia and thrombosis of this cutaneous vessels and ulceration. Sometimes you open up some of these animals that are septic, and you'll see granulomas granulomas in the liver, like we see here. In this particular case, this was due to salmonella. This was an animal that, reptiles have been implicated as being carriers of salmonella, and they are quite often. Salmonella Arizona complex can occur without any symptoms in their intestinal tract, but then if they get stressed due to parasite loads or poor husbandry, the organisms can go septicemic, or the animals can become septicemic. You'll see something like this. And sometimes these lesions can be quite extensive. This is a liver that is almost totally replaced by large kind of necrogranulomas, coalescing areas of necrosis. This happens to be that Matamata turtle I showed you a couple slides ago, same turtle. And what we cultured out of here was Citrobacter frundii, the same thing that you could see, was it proliferative colitis in mice. And that, when they originally described SCUD, the septicemic cutaneous ulcerative disease, they said it was due to Citrobacter. But now, that, as I said, that's really, it can be due to any kind of organism, and the name SCUD is not used. But not all bacterial infections have to end with the acute septicemic process. They get subcutaneous abscesses, granulomas, just like any other animal. One thing to remember is that due to the enzyme components of the heterophils in reptiles and birds, that they don't produce the liquid pus like you see in mammals. It's more of a granulomatous, or a caseous process. And so you open this up and you get this very thick, cheesy-like stuff out of there. In fact, when I took this picture, I thought this was a tumor. I thought it was a neoplasm, but then I opened it up and turned out it was just a granuloma, or due to, well, I guess we cultured Pseudomonas out of there. Talking about having abscesses, this is a eastern box turtle, and something that occurs quite commonly in wild box turtles, that if you, for any reason, you should happen to be in a practice situation where people are bringing in wildlife, I guarantee you're going to see this sooner or later. It's middle ear abscesses, very common in these guys. There's been a theory, it's something about the anatomy of their eustachian tubes. It's quite easy as they're eating for food and bacteria to migrate up the eustachian tube and go into the middle ear. And this is the tympanum that we're looking at right here. And you can see it's this bulging out. And if you incise it, then you pull out this kind of cheese ball looking thing that it's very typical of what is, I guess we could say it's reptilian pus. It's this real thick caseous material. The nice thing about it, it's kind of amazing if you do that. You can see this guy, this is the same turtle. But see, here he is. This is very painful. His eyes swollen shut. He's not happy at all. As soon as you open that up, I mean, it's almost like they smile. You go, thank you. And the amazing thing, you flush it out with a little iodine, and that thing will heal up within 3 days. I mean, it's amazing. And you'll see sometimes people will bring them in, and they've already started to rupture on their own. And I've watched these guys take their front feet and pull at these and actually start pulling this stuff out themselves. So I guess it's some kind of evolutionary advantage to be able to do that. Okay, here's a disease you better know about. If you don't see any other slide on the boards, you're going to see a snake with an open mouth. And you see a snake with an open mouth, the knee-jerk reaction is necrotic stomatitis, or the common name for it, the layman's term is mouth rot. This is one of the early cases of it. This is more of a hemorrhagic, kind of a necrohemorrhagic stomatitis. And the answer that they want, what is it caused by? The board committee wants you to say Aeromonas hydrophila. In my experience, it's almost always Pseudomonas aeruginosa. But again, it can be due to just about any gram-negative organism. It's almost always gram-negative. I don't think I've ever seen it due to a gram-positive. But they just get this overwhelming gram-negative overgrowth in their mouth for whatever reason, and you get the early stages. It's acute and hemorrhagic, and then later on, it gets to be a little more mucopurulent, and that's because because the mucous membranes are inflamed. You get a lot of mucus production. I told you that their heterophils don't produce liquefying enzymes, but if you've got inflammation of a mucosal surface, then you can get the mucopurulent form. It doesn't just have to occur in snakes. It can occur in any kind of reptile. We've seen it in turtles and lizards, and sometimes it can be quite extensive. As we see, this is a Komodo dragon. It's kind of a weird angled picture, and the brain's already been removed. This is the eye. But anyway, this animal actually, it had a disseminated neoplasm and was debilitated, and then finally what killed him was septicemia due to mouth rot or necrotic stomatitis. But that's my favorite gross out picture. The big worry when you've got an infection in the mouth is that it's going to migrate down into the lung. And this is a lung that's opened up, and you can see that this lung is just full of mucus and blood and purulent material, and all these bubbles are because this animal's trying to breathe through this stuff, and it's bubbling up, and I don't think you have any problem understanding why this animal died. So it's a common sequela to the mouth rot or stomatitis, just due to migration down of these organisms down into the lung. And the one thing about it, reptiles aren't able to cough at all. I mean, they don't have a diaphragm. They can't cough this stuff up. So the one big thing, the disadvantage that snakes have by having that long lung and long air sac is if they get something in the lung, they have a horrible time getting it out. So that's the trade-off. This is, in a more chronic case, you can have more of a granulomatous type pneumonia, as we see here. This happens to be a tegu lizard, 2 lungs here, that died with Pseudomonas pneumonia. And as you can see, you can appreciate it's much more of a granulomatous process. And this probably was more of a bloodborne infection rather than a bronchopneumonia like that snake. Just looking at it, it seems like the interstitium here is expanded. Okay, moving on to mycotic diseases. This is an Indian soft-shelled turtle. You can see that the carapace here, the dorsal surface of the carapace, there's a lot of ulceration, and you probably can appreciate in some areas it looks proliferative. This is due to superficial infection with a fungus. In these water turtles, you can get Saprolignia, just like you see in the frogs, or you can get just about any kind of waterborne organism. I've forgotten exactly what this was. It wasn't Saprolignia. This was a pigmented fungus. This was kind of more of a chromomycosis type thing. But I guess it was a phycomycosis. But anyway, there were 2 of these that were illegal imports, and the Fish and Wildlife Service confiscated them, gave them to us. The reason that they were illegal imports is because they're an endangered species, and no one had really kept them in captivity. And we got them, and I guess we weren't doing something right, because they both ended up dying with mycotic infections like we see here. In fact, one of these animals had it up here on the skin, and the fungus actually penetrated into its brain. So anyway, they do get superficial mycotic infections, much like amphibians do. Here's a snake, and again, this snake has had trouble shedding. He's got retained skin. One of the big problems with retained skin is it's a real good media for fungus to grow on. And at this point, he's got fungus growing on him. Mycotic cutaneous infections in snakes has been reported several times. And surprisingly to me, anyway, the most common organism that's been isolated has been Aspergillus species. Again, these are organisms that are in the soil, in the environment. So anyway, this animal was not cultured, but it was histologically, this was a mycotic dermatitis, proliferative. Okay, I apologize for the quality of this slide, but it's the only gross slide of this disease I have. We're now moving on to parasitic infections, and this is a longitudinal section of intestine from a lizard. I can't remember what species, and this is a cross section. And I hope you can appreciate that there's just diffuse hemorrhage and necrosis of the mucosa in this intestinal tract, and you can tell that the wall of the intestine is markedly thickened. There's a lot of edema and inflammation, and here's probably what they might call a flask-shaped ulcer, and that's kind of a key term. So this is amoebiasis due to infection with Entamoeba invadens, Invadens. Not to be confused with Entamoeba histolytica, which causes the problems in humans and monkeys. This is a totally different species that likes the cold body temperatures of reptiles. The interesting thing is that a lot of turtles can carry Entamoeba invadens asymptomatically. They're very, very common carriers of it, and this is one of the reasons that you don't want to mix your exhibits or mix species in the same cages, because turtles carry it, and snakes and lizards are extremely sensitive to it. And so the problem is you mix a snake or a lizard in with a turtle, and you end up the snake or lizard dies, and the turtle goes on quite happily. If the organisms are able to get into the bloodstream, they will migrate onto the liver. Here's a liver. This happens to actually be a turtle that died with entamoeba, but you can see that the large areas of necrosis, the so-called hepatic abscesses. That's what they call it in human medicine. And really, there's no abscess to it. It's just large areas of necrosis associated with organisms. There's usually not enough time for the inflammation to come in. So that's necrotizing hepatitis due to entamoeba infection. Okay, this is the stomach of a corn snake, and you can see there's a little bile staining down here, so this must be the duodenum. This is the esophagus, and you can see that the stomach is markedly enlarged, swollen. At this point, you might think, well, maybe there's a rat or a mouse in here, and that's why it looks distended. But when you open it up, actually, it's distended because there's been a marked proliferation of the mucosa. And this is due to infection with Cryptosporidia. I think you probably should know about this disease. You should know about entamoeba, about amoebiasis in reptiles, and you probably should know about Cryptosporidia. It's a fairly common disease in snakes. And the classic gross lesion is a proliferative gastritis. And the reason that it's proliferative is that the cryptosporidial organisms attach to the surface of the mucus cells of the glands, and then there's this marked proliferative response of the mucus cells. Mucus cells proliferate. The mucosa can be like 10 to 20 times as thick as normal, and it's all mucus cells. And so you can tell there's a lot of mucus in the lumen here. And at the same time there's a mucus cell proliferation, there's atrophy of the enzyme secreting cells, the enzyme and acid secreting cells, so that the animal is not able to digest its food. And the classic clinical signs are you've got a snake that's hungry that will readily eat, but then 3 or 4 days later it regurgitates this putrid, horrible smelling mouse or rat or whatever you gave it, and it's because the food item just sits in here and rots until the animal regurgitates it back up. And it's because they are unable to digest it. And so they end up dying either due to starvation. They literally just starve to death because they're unable to eat and to digest food, or then they get stressed and then secondary septicemias, mouth rot occurs, whatever. So anyway, this is a pretty classic disease. The species of Cryptosporidium involved is not well established. They know that it is definitely reptilian. It doesn't infect mammals. There's been experimental studies, and it's, the morphology, the size, everything is just different. So if you just say Cryptosporidium species, you'll get the points. Okay, well, as I said, that reptiles are intermediate hosts for a lot of parasites as well. This is you can see this is a coeloma cavity opened up. These are intestinal loops, and all these nodular things are encysted parasites. And maybe you can appreciate that some of these look more thin-walled and like they might have a little white spot, like a cysticercus. And in fact, these are, there's two types of parasites encysted here. There are larval cess toads and there are larval trematodes in here as well. So this was a mangrove snake, and again, I don't know what these are. I suspect that the final hosts must be something that preys on mangrove snakes, something like some big birds or something in Southeast Asia. But just about every wild-caught mangrove snake we've ever looked at has been loaded with these things, and they're totally incidental findings. They're just sitting there waiting for something to eat the snake so it can complete its life cycle. All right, here is a snake, and there are multiple little black spots here on the skin, and these are mites. The mite, if you look at them under the microscope, you can tell they're mites because they have 8 legs. And these are, this genus is Ophionissus. It's O-P-H-I-O, then N-Y-S-S-U-S. Nutritious is the species name, Ophionissus nutritious. These are blood-sucking parasites. In heavy infestations, they can actually lead to anemia and even kill the animal. They're also important vectors of rickettsial disease, and as I alluded to earlier, they think they may be able to transmit the boid inclusion body disease. So it's not at all uncommon to see them on snakes, and they can really build up to high numbers in a captive situation if you're not changing the cage frequently. You can kill them off with ivermectin, though, so there are ways to treat it. Okay, this is, again, a poor slide, but the only one I have, of a lung opened up, and you see this segmented parasite sitting here in the lumen of the lung, and this is a pentastome. Snakes are definitive hosts for pentastomes. In fact, crocodilians can be definitive hosts as well, and it's usually not a problem in snakes. Now, in very heavy infections, yes, they can lead to pneumonia and actually kill them. They do attach to the mucosa. You can see this little reddened area where this thing's been attached, and I think they mostly just feed off secretions. They're not much on blood sucking, but probably the most important thing about them is that it's not at all uncommon. If you're looking at wild-caught monkeys, you'll see larval pentastomes, and I hope Dr. Gaskin shows you later on today some larval